recording. And it is Tuesday, January 19th, week 3. What is due today, of course, are week 2 items, which is task 2, discussion 2, and your lesson 2 case study. And what are we doing today? Week 3. So we open up task 3. All right, either just thumbs up or just in the comment section. Does everyone see my screen? Are we good?
Yes, we're good.
All right, good. Thank you, sir. All right, so what are we doing this week? And again, to continue the theme that we started in week 1, which is, atoms make molecules, molecules then make cells, cells make tissues, tissues then make organs, organs make systems. So if there's something wrong with what's inside the cell, let's say, for example, this bubble thing is the cell, then it's obvious then that the tissues will have a problem. And then if the tissue has a problem, then the organs, and then so on and so forth. So we're going to be talking about cells and what's in them. And if you look here at task 3, there's some nice notes here that could supplement. And again, I like these notes because they're quick and it's easy to be highlighted. So there's a lot of highlighted words here, and it's just quick. And this chart right here is essentially what we're going to be going over today, which is the organelles, which are the kind of organs, mini organs that are inside the cell, what they look like and their function. And isn't that anatomy, which is the structure, and how they all work together, which is the function. So this chart right here in these notes are really, really good and really neat. And then we're gonna also talk a little bit about diffusion, osmosis, and how do cells things in the cells and outside the cells, how they go in and out of the cell, because we're going to be talking about the cell membrane or the cell wall. It is selective. There's certain things that can go in and certain things that can go out, and that's the function of the cell wall. We'll talk about that a little bit more later. Now, there's different types of cells. Let's see this one. This is nice, prokaryotes versus eukaryotes versus viruses. Again, more supplemental notes, and we'll touch on that as well today. So the big thing is, what is the cell made out of? What are the organelles and their functions? So that's the big thing today, and that one big chart pretty much sums it all up. And let's see, does it mention any specific chapter in the textbook? No, it doesn't. So let's go through, you know what? This is what I'll do for you. Let's have this chart up. Keep this chart up. And then I'm going to open up a classic picture of a cell. What's a good picture that has all the juicy parts? This one's nice. I can take this, minimize it a little bit, so move it over. And then you have this table here. Okay, so let's start here in the middle. This is a typical cell, animal cell. We're considered animals, right? And or mammals. And our cell is a little bit on the complicated side, but if you look at the cell, it kind of mirrors, that's why they call it organelles, because all these little structures inside here mirror your brain, your kidneys, your lungs. So they do things. And the first one that we're going to show is this right here, this big purple thing in the middle, and it's it's just colored arbitrarily, different colors. So this purple thing, this is the nucleus. And if we look here, did they mention the nucleus? No, they did not. So the nucleus, that's the control center, okay? And the nucleus is so important that it has its own skin. It has its own cell membrane, a nuclear membrane. So you could see here, there's like, it's like an orange, like, there's a covering. And you could see there's pores. So this covering, this nuclear membrane, is also selective. It allows some things in and allows some things out. Now, if you look inside here, the material that are out here, and if you could kindly, whoever has, if you can kindly mute so that there's no background noise. And if you have a question, I'll be monitoring the chat as always. All right, let me write down the next name. So the nucleus is, there you go. I'll do it myself. So the nucleus is, the brain of the cell. It tells the cell what to do. And inside the nucleus here, if you look at here, is DNA. DNA, deoxyribonucleic acid, is the blueprint of who you are. It has all the instructions on what the cell should be doing and how the cell should be communicating with other cells to do its job. So let's say, for example, this is a heart cell. It is the DNA that tells the cell what to do and what to be. Let's say this is a white blood cell. Well, the DNA tells it what to do. So the whole entire thing is the nucleus. You have a covering, and that's your nuclear membrane. Now, it goes inside here is the DNA. Now, DNA is a ton of information. It is a ridiculous amount of information. And next week, when we talk about DNA and RNA, you'll see how much information it holds. In all honesty, it holds everything. When is this cell going to live? When is this cell going to die? How is this cell going to function? What color are your eyes? What kind of, and even now we know from research in the last 10 years that even behavior is encoded into your DNA, okay? So the DNA, right? And the apt definition of DNA is what? Deoxyribonucleic acid. It is the blueprint of who you are. And what's a blueprint? It is the plans of everything that this cell is going to be and what's it going to do. And we're going to talk about next week the RNA, which is different. It communicates with this thing here on the outside of this. So if I have a picture like this and I point at it as a whole, that's a nucleus. The covering is the nuclear membrane, and inside here, this loose DNA here is called chromatin, chromatin. Now, within the nucleus, you have this tightly packed DNA called your nucleolus. The nucleolus, that's the storage form of DNA. Remember we talked about hydrogen bonds and how they're very flexible and they're very loose. Well, that's why we have hydrogen bonds, so we can tightly pack all the information and then uncoil it here into the chromatin when we need it. So you have your DNA, your nuclear membrane, which is this covering. You have the chromatin, which is the loose DNA here. And then you have the nucleolus, which is the highly concentrated DNA in the middle. And again, the function of the DNA is the blueprint of who you are or who this cell is going to be. Now, if you look at this like beige brown thing here, and you could see it has all these little bumps. That is your rough endoplasmic reticulum, or your RER. And right here on our chart, if you look at it, RER, it's processing and transport of proteins. Now, who tells this factory, the rough endoplasmic reticulum, what to do? Well, the DNA does, and they tell it through RNA, which is ribonucleic acid. So the DNA is the blueprint of who you are. The RNA sends that blueprint, encodes it, sends the message to this factory right here, which is called the rough endoplasmic reticulum. Now they call this part the rough endoplasmic reticulum because it has all these little bumps. These bumps are called ribosomes. The function of the ribosome is to create proteins. Proteins are made up of amino acids connected by a peptide bond, and they're in a specific order, kind of like a specific chain of pearls. And again, back to the DNA. The DNA dictates that specific order. So based on the order, it would make different proteins. And we've already talked about proteins when we talked about enzymes and how to speed up reactions. So all of that is produced right here in the rough endoplasmic reticulum. The rough endoplasmic reticulum, you could look at it like as a protein factory. And who are the protein workers? The workers that make up protein is these little bumps called your ribosomes. They could either be free, floating around here in this thing we're going to call cytoplasm, this jelly-like substance here colored in like light blue, or they could be attached here to the rough ER. Now, if you noticed right here, the endoplasmic reticulum here is smooth. That is your smooth ER. So if this thing here, the rough ER, makes proteins, the smooth ER makes lipids and carbohydrates. Because remember, we had three parts of organic elements that we talked about last week. Well, actually, there are four. The fourth part is your DNA and RNA, but you have proteins, which is what the ribosomes make, and this smooth endoplasmic reticulum that doesn't have any bumps, that makes lipids and carbohydrates. Doesn't that look like a good question? Rough ER versus the smooth ER. So rough ER, think ribosomes, think proteins, and smooth ER, think lipids and carbohydrates. The next one on this list here is the Golgi apparatus. Now, do you see these little bubbles that are floating around here, like this one and this one? Things have to move around in the cell. So in order for things to move around, it has to be packaged. It's like in your house. You can't have things just floating around in your house. Things have to be put in drawers and have to be put in boxes. So the function of the Golgi apparatus is to help process, and the big thing is to package and transport the proteins and also other items like garbage, or because you don't want it floating around here. So this is the Golgi apparatus, and it's known as the post office. It packages items for the cell, and it sends it to specific places. And isn't that what the post office does? It packages the thing that you want and sends it to a specific place. And that's the function of the Golgi apparatus, which is right here. And you could see that it's a little different from the shape of the rough and smooth ER because it is smaller, and you could see how it pinches out and bubbles out. And you could have things like this here, lysosomes, peroxisomes. So what do lysosomes do? Intracellular digestion. Sometimes we have to eat things and we have to digest it, and so does the cell. It eats things and digests things, and some of it is garbage. And you also have peroxisomes, which does the same thing for fatty acids. Now, these lysosomes and peroxisomes have a bunch of enzymes that break down stuff. So you don't want that stuff floating around in the cytoplasm. So that's why the Golgi apparatus then packages all that. So this lysosome doesn't spread all those enzymes and break everything down, okay? Which, by the way, when this cell dies, there's signals to the Golgi to do what? Open up all these packages so that the cell can die and it can be digested from within. So lysosomes, think intracellular digestion, breakdown of things. Peroxisomes, intercellular digestion, specifically of fatty acids. And they are all packaged by the Golgi apparatus here. Now, this whole thing needs energy. It needs, the cell and your body need to be powered. And we already know that we consume oxygen. That's important. We also consume food so we can create glucose. Well, there's a reason why we do those two things, why we breathe. That's called respiration, or the exchange of oxygen and carbon dioxide. The second thing is why we eat, because I need to break down whatever I eat into pretty much a 6-carbon sugar called glucose. And all of that goes into this thing called the mitochondria. The mitochondria is the powerhouse of this cell. It provides the cell with energy, and that energy is called ATP, or adenosine triphosphate. Let's look at ATP. If you look at adenosine triphosphate, here's the chemical formula of it, right? It has a adenosine group, and it has 3 phosphate groups, if you see here. Now, ATP is awesome because remember we talked about bonds, and bonds typically are in a low energy form. Well, phosphate bonds are not low energy. They are high energy bonds. So ATP is your metabolic fuel. It is derived from taking in oxygen and glucose, and it makes this thing. And every time your body hydrolyzes or cuts this bond, right? And guess what we cut it with? We cut it with water. That's why it's called a hydrolysis reaction. Every time we cut this bond, right? So ATP will then turn into ADP, or adenosine diphosphate. If I let go this phosphate group right here, right, what will happen? Because it will release energy. And that's what the cell uses to do what it needs to do, because all of these processes require energy. So the powerhouse, or the power production of this cell and living organisms, is the mitochondria, which I believe is the point of discussion on your discussion for week 3. So your mitochondria produces ATP and requires oxygen and glucose. And we breathe oxygen and we eat food to make glucose. Next, centrioles. Well, let's talk about microtubules first, and then talk about centrioles. So this cytoplasm, this gray thing here, there's these proteins that act like a scaffolding. If you see all these little lines here. And what is a scaffolding? A scaffolding is like a structure that, you put on the side of a building so it does maintenance, and also it helps, it helps hold up the shape of this cell, because all cells look different and have different shapes and have different shapes for different functions. We already talked about that in week 1, right, when we looked at histology and microscopy. So all these microtubules here, they're all dispersed all throughout this thing, this jelly-like substance called the cytoplasm. Another thing that these microtubules do is also form some sort of like internal highway. So remember we talked about those lysosomes and peroxisomes, the little stuff that got packaged? Well, there's these little lanes or little highways that can travel. Now it gets to this idea of centrioles. Sometimes cells double up their DNA because they need to divide. 1 cell becomes 2, 2 becomes 4. It's essentially how you started off, all of us started off as 1 cell from your mommy, 1 cell, goes 1 sperm from mom, from daddy, 1 egg from mommy, and then we multiplied. And the one way we do it is these things right here. See these 2 things that look like little sprockets? Those are centrioles. Now, what do? When it's time for this cell to divide, the centrioles, one will move on one end of the cell, the other will move on the other end of the cell. And what will it do? It'll bind to all of these microtubules and then form these little like lines. And then everything that gets duplicated will get separated on one side and on the other as equally as possible. And that's the function of the centrioles. It's organization and movement goes of the genetic material, right, in the form of chromosomes during cell division. And the centrioles give it that organization. They start pulling the cell from side to side and then organizing the microtubules in here. All So let's look at a picture of that. What does that look like? Because it's one thing to explain it. Let's look at what kind of picture. So that's a centriole, which is essentially a whole bunch of coiled proteins together. But I wanna find it in cell division. There you go. Here's better. See these are chromosomes. They're representative of genetic material, right? These centrioles, right? They move, one moves to the one end of the cell, the other one moves to the other end of the cell. And see, it forms these little highways, these little scaffolding and tubing, so that genetic material will then be separated equally, so that when this thing splits into two, you're going to have one set of genetic material over here and another set of genetic material over here. It is highly complex on how it does it, and it's kind of neat on how it does it, because it it organizes the movement of the chromosomes or the genetic material during cell division. Another thing that proteins do, they form cilia and flagella. Cilia are like the hair-like. Let's look at another picture. In the cell wall, there's these little hair-like things, and they also use it for movement. They also use it for protection. Maybe you've seen this before. So they are pretty much a projection of this cell wall. Same thing with the flagella. Now, flagella, it forms like a little tail, kind of like sperm, and then it swashes around so that it provides movement. So there's two different types of movement. So cilia, think like hair-like processes around the cell, and flagella, think some sort of like tail. And the word, if you've ever heard the word flagellation, flagellation is like a whip. So you know when you whip yourself, if you're into that kind of thing. So it has for cell movement, because sometimes the cell as a whole has to move around. Microfilaments, nice to know. Intermediate filaments, nice to know. But microtubules, that is really important, because these microtubules not only form those highways and the scaffolding within the cell, it also forms the components that make up the cilia and flagella, which is an extension of your plasma membrane. So let's now look at this plasma membrane a little bit more closely. Now, what's the plasma membrane? This thing right here. Okay, it's the covering of the cell. The nuclear membrane, this thing right here, is the covering of the nucleus. But we also have a covering of the cell as a whole, all the way out here. So let's look at a picture of that, which is and the classic picture is this one. I always use this one. Khan Academy has a very nice picture. So if you look at this, that's what the cell wall is made out of. It's made out of a mosaic of phospholipids, and it has a bilayer, or two layers. And if you look at it, the outside, see these phosphorus heads here? The outside is hydrophilic. It means it loves water, right? This side too. See where all the little circles are? Those are the phosphorus heads on a phospholipid bilayer. Do you see how it's two layers? You have this one top layer, which is to the outside of the cell, and you have this bottom layer, which is now the inside of the cell. And so it is a phospholipid bilayer. And what do we know about phosphorus? That it loves water. So this orange outside, hydrophilic, loves water. This orange here on the inside loves water. And it makes sense. There's water here on the inside of the cell, and there's water here on the outside of the cell. So that's a classic question. What is your cell wall made out of? It's made out of a phospholipid bilayer. So the phosphorus heads, and then here you have your fatty acid lipids. Now, what do we already know about lipids or fats is that they're hydrophobic. They don't like water. So it forms like a nice insulative barrier here. So whatever's on the outside stays on the outside. Whatever's on the inside stays on the inside. So what are some features of this wall? And you can now see that it's a barrier. Things don't get through here. Oh, which by the way, again, I'm jumping on my soapbox again. A virus can go through this wall. That's how small it are. Let's see how small a virus is. See the dot on the I on this word lipid? That's about the size of your typical coronaviridae, which by the way is SARS-2 COVID. So begs the question, why do we wear a mask? And we're gonna and hopefully if you heard me before, we know the answer. But the answer is not because to keep a virus out of your system. If this like 12 kilodalton thing, this about the size of this little dot, can get through here, what do you think that the 10-cent mask that you're wearing, that I'm currently wearing? And remember, there is a greater reason and a better scientific reason why we wear PPE, but it's not to keep a virus in or out. So how do we keep things in or out? Even a virus, right? Again, we have the bilipid layer, okay? And we also have an immune system that monitors everything on the outside, okay? It also monitors things on the inside. So if it sees something that's foreign that it doesn't like floating around, well, I'm not sure if we already talked about that, or we will be talking about that in greater detail in anatomy and physiology too, is that's how this bilipid layer also stops things. Now, how do you know that a cell is a good guy or a bad guy? So do you see these things that are sticking out of here, these green things and these blue things? Well, they are glycolipids. Glyco meaning sugar, glycoproteins. Glyco meaning sugar, and protein, of course, a bunch of amino acids that are strung together, and that's why they have this in these little dots. So those are little amino acids. So your glycoprotein layer is like a coat. I'm wearing my lab coat right now, and it identifies me as what? One of the medical people, right? You wear the white coat, you're a medical person. So all the cells, right, have this glycoprotein coat. Even, and we saw, we all already know what this coronaviridae look like. It looks like what? A tennis ball with little bumps on the outside of it. And those bumps on the outside of it are the glycoprotein coat. And then when they're talking about, ooh, the scary variant, the coat changes sometimes, and that's why it gets harder to fight it, because you recognize the bad guy wearing a black hat and a black coat, and you're like, ooh, that's the bad guy. Well, the guy could be, because the coat could change to gray, and then your immune system will not recognize it as quickly as it should, and that's why there's a lot of hype right now regarding the variant. But those of us who know a thing or two about viruses it is an eventuality, and mutations occur all the time. In the last 30 years, in our study and our fight against HIV, right? The code already changed twice, significantly, right? So it's something that medical science and virology, which is the study of viruses, already know that this thing here, this glycoprotein coats right here that are sticking out, sometimes they change, okay? And it's lovely because they also form, see these protein channels here? These big blue things are large proteins. And some of them form little channels. And these channels have doors. And guess what these doors are made of? They're made out of glycoprotein. So let's say, for example, I have a very large 6-carbon sugar like glucose that wants to get inside the cell. It's big. It can't go through this wall, right? This wall is surrounding the entire cell. Well, we have insulin that comes in and it fits on a receptor, which is on which is on your glycoprotein coat. And then what will it tell the channel to do? It can tell the channel to open it up so that glucose can go inside the cell and then be utilized by the mitochondria to make ATP. And that's why diabetes mellitus is so insidious and so dangerous, because if all my fuel is here on the outside of this vehicle or outside of the car, don't you think it can do damage? Where does the fuel need to be? It needs to be inside the cell, inside the mitochondria, so we can make ATP. And again, the analogy for diabetes mellitus is it's just like going to the gas station and pouring the gasoline all over your seats and all over the hood of your car instead of putting it inside the gas tank. And that's what insulin does for us. It's a chemical messenger, sits on a receptor, glycoprotein, that's on the phospholipid bilayer of your cell, and it tells the protein channel to open up so that the big molecule, glucose, can go in. Now, here's the neat part. Don't need insulin to get sugar inside your system. If you work out on your regular basis, right, or you're, especially if you're living this healthcare sedentary lifestyle, move around, walk around. Um, I'm on campus today, so I'm sitting, but when I teach my evening classes, I now walk on the treadmill. I set up a stand, and so that at least I'm burning something, especially on the days that I can't get to the gym. But if you get to the gym, there are other doors or other protein channels. They're called GLUT2 and GLUT3 protein channels that open these doors so that the sugar stuff that you ate, all that here, can go inside and can be utilized. So these glycoprotein coat is good for two things: immunology and for opening and closing these protein channels. You also have these proteins here. You see these stones that this is a fluid mosaic model, so it's kind of like wavy and kind of flimsy. So you put these proteins in between it, and it makes the wall a little bit more rigid. So you have these what they call integral proteins, like a protein like this, 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 and this, that gets interdispersed all throughout this phospholipid bilayer um, um, to make it, stronger. Okay. Now, see this? These little hexagons here? That is cholesterol. Now, what is cholesterol really? Cholesterol really is a vehicle that moves fat in or out of the cell. So that's why laypersons always talk about the good protein, the good fat or the good cholesterol, which is HDL, high density lipoprotein. HDLs are the cholesterols that are embedded within your phospholipid bilayer or your cell wall that allows fat to go where? Out of the cell. Now you have way more cholesterol in the form of your VLDL and LDL, which is low density lipoprotein, very low density lipoprotein, lipo means fat, protein, we already know what a protein is, and that's what cholesterol does. It brings things in and out of the cell. So if you modify your diet, right, and have more HDLs, right, a little bit more HDLs that'll carry the cholesterol out, then that's a good thing. So it will kick out, fats. But if you have a high diet and fat, you're going to have more vehicle cholesterol in the form of VLDL and LDL will bring fat in. And all of this is part and parcel of this phospholipid bilayer. So if we look at that picture, it's just a covering, but if you look closer, it's way more than just a covering. It is a gate. And also it is known as the gatekeeper. It will allow certain things in and it will allow certain things out. Okay. And since it forms like a little puzzle, that's why they call it a fluid mosaic model right here. Now, there are some rules on how things go in and out of this cell. And one of those rules is called diffusion. Like, for example, we know how diffusion works. Let's bring this to the forefront here. And these are your notes from task 1. If you go into the, let me go back here. Task 1, task 3. If you go to task 3 and you click on self, structure, and function, this is task 3, and you click on this thing right here. There's some quick notes here. And there's a lot of what I was talking about. There's all in here. Nice chart. Phospholipid bilayer, right? We have these peripheral and integral proteins. We talked about that, and their function is to make the phospholipid bilayer stronger, and that's why they call it a fluid mosaic model, because if you've ever seen a mosaic, right, it's art that it's art that's made up of all these little pieces, and that is what your plasma membrane is about. So let's talk about diffusion and the concentration gradient. I'm not sure when's the last time any of you have been in a mall, but it's been a while, hasn't it? But when you go to you go to Macy's or Nordstrom, and you go into the perfume counter, right? When that lovely lady, sprays the perfume, right? When you spray it right there at the table, it's kind of strong. And then the further away you move from the table, the strength of that perfume gets weaker and weaker and weaker. And that's why the perfume kind of dissipates and kind of separates out, because it wants to move. The perfume wants, and all the molecules of the perfume want to move from an area of high concentration, which is at that desk when the lady sprayed you, to an area of low concentration, which is the rest of the mall. That is also called entropy. It's a thing in physics where everything in this world wants to fall apart, wants to get to spread out. It's like you put powder outside and there's some wind. If you leave that powder, outside on the ground, it's eventually going to do what? It's going to fly into a million particles, and it will, all intents and purposes, be so diluted it will disappear. So that's what your body wants to do all the time. It wants to move things from an area of high concentration to an area of low concentration. And that is called diffusion. Okay? And the concentration gradient is, in order for diffusion to happen, it requires no energy. Okay? And the steeper the concentration gradient, for example, if the concentration gradient right here is very, very high and it's very, very low over here, what's going to happen? The diffusion, or the rate at which the molecules are going to spread out, is faster. Temperature also affects things. It moves molecules faster, right? And we know that because you leave meat or milk or anything that gets exposed to the air outside, what's going to happen? Its molecules are going to start to break down and it's going to get rotten. But if you put it in the refrigerator, it lasts a little bit longer. So the colder the temperature, the less movement of molecules, it will prevent diffusion. Surface area is also important. It's also the reason why your lungs separates out into a whole bunch of branches. So there'll be greater surface area. There's also a reason why your intestines are all highly coiled, especially your small intestines, so that has increased rate of, increased area, so that increased surface area, increased rate of diffusion, so that all the food, when you eat it, and all the the water, when you drink it, can easily diffuse into your bloodstream so that you can reap the benefits of what you just ate and drank. And last but not least, if it's a larger molecule like sugar, it's going to move slower. A smaller molecule like an ion, like we mentioned in the last lecture, like lithium or sodium or chlorine, it'll move a lot faster. And that makes sense. Something big is going to move slow. Something very small is going to move very fast, right? So let's say, for example, you have a small, very small, very fast drug like lithium. It moves through your system very quickly. But something like glucose, especially for the diabetic patient, they're large molecules, they move really slow, and it tends to mess things up. Now, diffusion, again, is movement of molecules from an area of high concentration to an area of low concentration. And in order to move up the gradient, right, from an area of low concentration to high concentration, which your body does for other things, it requires energy. But moving down the gradient from an area of high concentration to an area of low concentration does not require energy. So let's talk about osmosis, because our whole entire body has these cells. And these cells, for all intents and purposes, are permeable membranes or selective semi-permeable membrane. So let's talk about these kinds of solutions. You have isotonic, hypertonic, and hypotonic. So isotonic, if you look at the prefix, means the same or the same tone. That means the two, if you have, you can pretend that this is a semi-permeable membrane and there's two cells in communication with each other. If you look in an isotonic solution, you know, let's say these little like white dots are water, and let's say these black dots are like salt. So if you see here, 1, 2, 3, 4 salt molecules, 1, 2, 3, 4. So, and how many water molecules? 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So we can say both the left and right sides are equal, right? And they're isotonic because they both have the same concentration of water and the same concentration of salt on either side. But let's say, for example, you have two different cells. One will have more. This one has way more salt. 1, 2, 3, 4, 5, 6. So this would be a hypertonic solution. And this only has two salt molecules, this one and this one. And that will be a hypotonic. And remember, hyper, above or too much. So hypertonic solution, hypotonic solution. And the neat thing about osmosis, right? Your body is constantly moving these molecules around, both water, which is this thing, and salt, because its goal is ultimately what? An isotonic solution. And in the human body, it is normal saline, or NSS, normal saline solution, and that is 0.9% sodium chloride. Okay? Know that, because that's a very, very common thing. And if something's higher than 0.9%, it's hypertonic. If something's lower than 0.9% sodium chloride, then it's hypotonic. And remember, if you have two things that sound alike, like this thing, memorize one like your life depended on it, and the other one is simply what? The other. Now, what's the important about these things, osmotic pressures and, you know, the salts? Because these things are important because wherever salt goes, water has to follow. So if we have a semi-permeable membrane here, and that's what this experiment that almost never worked. Back in the day when I was at Falls Church campus, I think I did this experiment like every term for like 2 years, and it never worked. Well, in theory, if you have a semi-permeable membrane and you put more salt in one side, what happens if I put more salt in the form of these big black circles? If I have these larger black circles here, right, there'll be less water molecules here. If there's less water molecules here and more water molecules here, there'll be an osmotic pressure gradient, right? More water here, less water here. That means where will the water go? It'll move through this semi-permeable membrane to this side. And that's essentially how your kidney system works. When your kidneys start releasing salt, the water in your system will follow it because of the osmotic pressure. So more salt, the water will tend to go wherever the salt goes. And you'll learn a little bit more of that in your biochemistry class. But just know that wherever salt goes, water goes. So if you have a diuretic, which is your water pill, I give my patient a diuretic. The diuretic pill or the diuretic medication tells your kidneys to start throwing out salt. What will happen? Your kidneys will also throw out water. That means you'll urinate more. If you urinate more, what happens to your blood pressure? It drops. And that's why we use diuretics, to drop your blood pressure. But it's annoying because every 15 minutes, your poor patient has to go pee. And this is how we play with hypertonic, hypotonic solutions. And also, this is also how we know that the human being is made up of 66% water, which is 0.9% sodium chloride solution, also known as your normal saline or an isotonic solution for human beings. All right, and that's the end of those notes. And let's go back here to our task 3 to see if we talked about everything that we needed to talk about. So did we talk about the cell membrane? Yep. Cytoplasm? Yep. Cellular organelles? Yep. I'm going to hit up this tutorial in a minute. And remember, the notes are here. Do you have enough to make a concept map of cell structure, organelles, and the function of organelles? Yep, pretty good. So let's look at these types of cells. You have prokaryotes, eukaryotes, and viruses. So if you run through this thing, what are prokaryotes? You click on that. A prokaryote are like bacteria, okay? They're similar to eukaryote, which is you, the human being, but it's a little bit different, okay? It's more simplistic. So the basic difference between prokaryote and prokaryote, I want you to think what? Bacteria, okay? And think that there is no such thing as bad bacteria or good bacteria. Now, the bacteria that you need to survive in your body, that's called normal flora. Flora means growth, right? So, like, for example, E. coli, is not a bad thing. You need it inside your colon to make vitamins to act as cofactors for your enzymes. That's why when you take an antibiotic, what is the most common side effect? You'll have stomach cramping and diarrhea and all that jazz, is because when we take an antibiotic, it not only kills the pathogenic bacteria that we want to kill, it's going to kill a decent amount of normal flora, and then it causes a whole bunch of problems. We're getting to that, sir. So bacteria is what? Prokaryote, okay? It is similar to our cells, but to the human cell, but it's simpler. Now, the eukaryote is you. EU means normality. We know that from our from medical terminology. So the eukaryote is you, the human being, okay? And it has all the organelles that we just discussed. So if I could ask you, what's a human animal cell? You'll tell me eukaryote. What is a bacterial cell? Prokaryote. Now, what's a virus? A virus is essentially a parasite, okay? And what is it? Let's see a picture of one. We already know what our cells look like, but this is what I was talking about. This is a typical bacterial cell, right? Which is a prokaryotic cell. And this is a rather large virus. And look at here. right? Look at HIV. Look at polio, right? It's tiny. It can easily go inside here, right? So then with it being so tiny, if you look at a virus, what's it made out of, right? It has this, some viruses have this little head and these little tails, or it could be what COVID looks like, right? But no matter what the virus is, inside it has either DNA or RNA that can utilize the host DNA or RNA. Now, see how virus invades. Let me see a good, maybe like this one. Okay. If you look at this, remember you have the cell wall, and the virus is very, very small. And you could see here, there's a single strand of, and that's RNA. So remember we talked about, we're going to be talking about RNA in greater detail. But what a virus does is it's so small, it gets in here, and it gets inside the nucleus, inside the nuclear membrane, right? And then you see this double helix DNA molecule? It takes their information and then it puts it in here. That means it hijacks the cell. And now the blueprint is now no longer the cell's blueprint. The instructions to go into the rough endoplasmic reticulum are no longer the instructions of the cell, the host DNA. It's now the instructions of the virus. And the virus is a very simple thing. All it wants to do is live and make more of it. That's all. It's not a good thing or a bad thing. It just wants to live. And that's why it's so insidious. And since it's so small, it's very hard to fight. And also, once this virus, you know, your immune system, it kills a decent amount of them on the outside. But once this virus is on the inside, you can't see it anymore, right? It's kind of like a home invasion. If you look at a home invasion, there could be horrible things going on inside the house, and the police, which is your immune system, would never know until it's too late, until the house is on fire, right? Which happens in all intents and purposes over a virus. And then now you have not one, but millions of these, and then they spread out. And that's why viruses are so insidious. They're parasitic. They don't need much to live. And they hijack your own DNA or RNA. HIV does the same thing. It inserts itself, and then instead of the cell making instructions to do the cell, it makes instructions to make more HIV. So let me make sure I got everybody on this call. 1, 2, 3, 4. Good. Because remember, for those of you who are not on the call today, the new university policy for this term is that you have to be at the Zoom sessions. And if you cannot be at this Zoom session, I need to get an email, I need to get a call. So please do so. And that's my little public service announcement. So that's what a virus is. It's very, very small. It doesn't need much to survive, and it can inject itself into, it will inject itself into the host nucleus, and then tell the host nucleus that here's the new plan, here's the new DNA. We're going to make more viruses instead of doing what you're supposed to be doing. And that's why viruses are they're very, very nasty, and they're hard to kill. We've known about the common cold, which, by the way, is the same exact family as COVID. It's a coronaviridae. And if you look at the shape of COVID, it's like a crown. And that's why they call it corona and coronaviridae, or coronavirus, a family. Oh, by the way, when you get sick by the flu, let's say you get sick today, right? You get well, and your immune system makes antibodies and remembers that you got sick today by coronavirus number 135. Next week, you could get hit by coronavirus number 137, and it will still make you sick, right? And that's why also, again, back on my conspiracy soapbox, something that small, I'm a chemist also, so I know of no technology that can tell me within 48 hours that it confirms you have a virus. Even when we test for HIV, it takes days. So what are these diagnostic tests made out of? And who's testing to see if it's efficacious or not, right? How do we know that these new people have the new variant? Because clinically, there is also no diagnostic criteria on what COVID is, isn't there? Right? It's not like HIV. HIV, you know, you have AIDS when you have pneumocystis carinii, you have immunocompromised disease like tuberculosis, you also have Kaposi sarcoma, and your CD4 cells are in the basement. There is no diagnostic criteria for COVID. So how do you know you got it when I can't trust um, uh, um, my subjective data, like interviewing the patient, right? Because from one person to another person, it's different. And also, I can't trust my objective data, which is the test, right? So just make you guys think a little bit, right? So how is this? And that's why a lot of people who are saying that stuff like, stupid things like this virus isn't real. It's real, and it's really killing people. But what we don't know is how exactly, and the preventive measures. So we have to do things that make also common sense. Now back to the PEP. Why do I wear my mask? I wear my mask because there are 249 other things that will make me sick. It makes me sick, it drops my immune system, and guess who gets to come in? SARS-2 can get to come in and do its damage, right? So it's at this point of the show, well, before we leave this lecture, let's go over what we need. So for this week, of course, for next week, of course, task 3, and that's your concept map, and also the discussion. Watch this video on mitochondrial disease, but you don't really have to watch a video on mitochondrial disease. You can also look it up, but discuss how the mitochondria, which is the powerhouse of the cell, how it relates to this disease, and make sure you grab a source. It could be your textbook, and again, have your opinion, your rationale, some sort of body of evidence, and a conclusion to wrap it up. Okay, and that's of course due next week. And lesson 3, lovely video here. Watch the video on lysosomes. There's this cute video here that I've seen better, but it's a nice video on how the ribosomes are protein production makers, hence the term rough endoplasmic reticulum. That is your protein factory, and the ribosomes are your protein factory workers. Do not complete the lab, but there's a nice cell structure and function, which I believe is the same set of notes, right? Repeated. And then you have an osmosis and diffusion lab. Don't do the lab, of course. But again, more repeated notes right here. So you have 3 sets of the same notes on 2 different locations. Now, what's due? You click on down here at the bottom of lesson 3. That's the location of your application assignment. You click on that, click on download the case. We look at this. You read the case, answer the first set of questions, 1 through 5. Read part 2, answer the questions 1 through 7. And then, of course, part 3 answers 1, 2, 3. And I think that should be good enough. Sections 1, 2, and 3, right? and those questions. There are about 11 questions in total. So section 1, questions 1 through 5. Section 2, questions 1 through 7. And section 3, questions 1 through 3. Okay? And that is due next week. Now, let's look at this, because this is a beautiful future NCLEX question. And then they talk about this genetics here. And if you could see, there's these letters. These are called your genotypes. Those are the letter representations of the phenotype, which is the physical characteristic of the person that they're discussing. So you have daddy here and mommy here, okay? Now, this is, let's talk about this with respect to sickle cell. Now, sickle cell, you could either be a carrier or have and the carrier also sometimes has mild symptoms. But the person who has the full-blown disease, they have severe symptoms. I'm talking about giving you morphine level of pain in sickle cell disease. But if you have the sickle cell trait, you could have mild symptoms or you could be a carrier. So let's say, for example, daddy is a carrier, mommy is a carrier, and it's represented like this. Now, a full-blown person without any sickle cell disease will have 2 capital letters right here, right? And you could see where he got 1 from his dad and 1 from his mom. Or you could be a carrier, which is you can have the dominant capital R, and that represents, goes that this person will be a carrier and they're just carrying the little r, which is the disease. And of course, the person who's fully affected has 2 lowercase r's, 1 from the dad, 1 from the mom. So if you have 2 carrier parents in sickle cell disease and this Lay syndrome, which is what the case talks about with that little baby girl who unfortunately passed, right? The odds of you having a child who has no disease, no carrier, is 25% or 1 in 4. The odds of you having one that is a carrier or with mild sickle cell trait is 2 out of 4 or 50%. And the odds of you having a child that has a full-blown disease is 1 in 4, 25%. So when you look at this, how does the disease get represented? This 2 smaller r's is called homozygous, meaning homo mean the same, right? Recessive, because they're recessive genes. They don't get expressed unless both of them are expressed. That's why these 2 children here, they are heterozygous. Hetero means different, right? With a dominant trait here that's hiding this recessive trait, the lowercase r. And then here, you have a homozygous dominant. He has 2 dominant genes, or she has 2 dominant genes. That's why this child here is unaffected. So what kind of questions come out on a typical NCLEX? If I don't show any phenotypes, or a phenotype is who has the trait or who has the physical attribute or who has the disease, that's a phenotype. And I just showed you the genotypes, the letters. You will now know if it's heterozygous dominant, this person's a carrier. Homozygous dominant, this person is fully normal. Homozygous recessive, this person will have the full-blown disease. And that is called an autosomal recessive inheritance. And just one example of genetics, and also one example how you get half of your genetic material from your dad, half of the genetic material from your mom, right? Human being has 46 chromosomes, 23 from dad, 23 from mom to make a whole human being. Daddy's sperm only has 23 chromosomes, only half the genetic material. Mommy's egg only has 23 chromosomes, only half the genetic material. But you put it together, you have 46 chromosomes, you have a full-blown human being, or full-blown what they call progeny, or your next generation and children. So this is That's a classic NCLEX question. Or they could give you the phenotype. They can tell you it's autosomal recessive inheritance, and you'll state like, oh, my patient has homozygous recessive for sickle cell. That means what? Full-blown disease. They have heterozygous dominant. That means they're a carrier. They're homozygous dominant. Oh, that means the child is unaffected. Or, and then they do stuff like, what would happen in the next generation if this person marries a carrier, or if this person marries a carrier, or if this person marries a carrier. You could see how that can be expanded as well. Alrighty, I believe that is our lecture. I have 4 people on the count. All right, so it is at this point of the show